Good morning, everyone. I'm Awaz Khan, a plant pathologist working at Cornell Agritech in Geneva. I'm really excited to present to you database of apple diseases and advanced disease diagnostics for orchard health management. Just want to give you a brief overview. There are a large number of diseases and pests which cause huge economic loss to apple production every year. These diseases are a particular threat to high density monocultures or susceptible cultivars, particularly in favorable environmental conditions of northern US. Growers need to use chemicals very frequently, which can result in emergence of pathogen strains which are resistant to these chemical sprays, leading to increased input cost. In addition, frequent use of chemicals have human health and environmental concerns. Now the question is, why do we need precise, rapid, and sensitive detection of diseases and pathogens? I will give you just a few examples briefly. If we have few spores of fungal pathogen, for example, apple scab or powdery mildew, and if there is favorable weather, and also if there is highly susceptible cultivar, in a very short time, these few fungal spores can multiply and become hundreds and thousands and millions, which can really devastate the whole crop in a very short time. Therefore, it's important to identify and detect this disease as early as possible. Another aspect is misdiagnosis. For example, here you see on right side fire blight. So fire blight symptoms sometimes can be misdiagnosed as spray damage or necrotic blight, or vice versa. We did survey of apple growers in New York State that included growers at Lake Ontario Fruit Tour and also Young Fruit Growers Tour in Highland. And we asked them about their current practices for fire blight identification. And most of them, they said that they are comfortable for visual identification of fire blight based on the symptoms. And also some told us that they send samples to Cornell labs for identification. And based on those results, I can tell you that we cross-checked what growers or extension educators were saying fire blight and what were the results from the Cornell lab. And there were many differences in many cases, indicating misdiagnosis. And just furthering the previous point I was making about this misdiagnosis, if there's misdiagnosis of fire blight, it can turn into a devastating issue for the orchard, but not only orchard, also the whole region, because the inoculum builds up, and when the conditions are favorable and enough inoculum and susceptible cultivar, it can spread very quickly and kill the orchard and many orchards in the region. With the financial support from New York Farm Viability Institute, we have been optimizing the use of two immunokits called AgriStrip and Pocket Diagnostics, as well as we have been developing a method called loop-mediated isothermal amplification, and this is based on DNA amplification. Here on this slide, you see both immunokits. On left side you see AgriStrip and on right side you see Pocket Diagnostics. These both are commercially available kits and they both have the same or similar principle based on antibody detections. In both kits you take the suspected sample which you think that might have fire blight and crush it in the buffer which is coming with these kits. And after crushing the sample, you take this uh, sample or liquid, either you put in a cup and put agar strip kit or agar strip, the actual strip into the cup, or you take a dropper and take the liquid and put the 1 or 2 drops of the liquid into this hole on this strip on right side from Pocket Diagnostic. Both of these strips have control and test lines, and if you have both lines visible, for example this blue line on Pocket Diagnostic on extreme right side, then it means you have your sample positive for fire blight. One can use different kinds of samples for detection of fire blight. For example, if you have a suspected sample, you can use leaves from the sample, you can use the wooden part of the branch, or if there's any ooze droplet, you can use that also to test. We have also compared the sensitivity and specificity of both immunokits. Our results show that Pocket Diagnostic Kit was more sensitive compared to AgriStrip Kit. Specificity of AgriStrip was better than Pocket Diagnostic. Pocket Diagnostic Kit was able to detect signal or positive signal in Erwinia species which are not responsible to cause fire blight. In addition to antibody-based fire blight detection, we have also optimized a DNA amplification-based method called LAMP. LAMP method can be divided into 3 steps. First is the DNA extraction from the suspected sample, then amplification of this DNA, and then finally detection of this amplified DNA. We have used a commercial device from a company called BioRanger, and this device is also linked with a tablet. So the amplification of the DNA happens in a small tube which has all the reagents to needed for the amplification, and we put that in this device, and this device provides the temperature for amplification, and then amplified DNA can be detected in real time through fluorescence detection in this tablet. Other methods are also there to detect this amplified DNA, which are based on color change, but we highly recommend using this battery powered commercial device connected with tablet. On this slide, you see the screenshot from the tablet showing the amplification curve for the positive and negative samples. You can clearly see that between 9 and 10 minutes, there is already the amplification peak going up for the samples which are positive, and the samples which are negative, the the line is pretty straight. For example, the sample in gray. So within 15 minutes is possible to tell that if the sample is positive and has fire blood infection or not. In conclusion, immunokits are more time efficient, but their sensitivity is lower than LAMP. They are also less specific compared to LAMP. However, they are easy to use in the field, and cost to test one sample ranges from $3 to $10, whereas LAMP can take longer time, especially for preparation of the samples and also for detection. Whereas the initial cost for buying this LAMP device and tablet can be high. Now I will move to another emerging opportunity for efficient management of apple orchard health, and this is based on use of drones. I will divide this part of my presentation into introduction of UAVs, sensors, capturing data, processing and interpretation of data, and finally, use of this data for precision orchard management. UAVs can be divided into two broad categories, multirotors and fixed wings, and it really depends on objectives, your needs, and the features of an orchard to choose between multirotors or fixed wings. For example, fixed wings can cover a large acreage in a short time and can be more battery efficient, while multirotors can be more efficient for customization to change the sensors and cameras, etc. Typically, a UAV has GPS, cameras and sensors, and battery. We also need a controller and smartphone or a tablet to operate it, and a calibration panel can be very important if you need to account for the variation due to sunlight or other environmental variables, and if you want to combine data from multiple time points or multiple years. Sensors are the most important part of a UAV. The principle behind sensor-based detection of diseases is the relationship between electromagnetic spectrum and biological processes. In this graph, you can see the relationship between the wavelength and different biological processes. For example, one can detect chlorophyll absorption and leaf pigments in visible light, whereas the cell structure and other vegetation reflectances can be detected in near-infrared wavelength, whereas water content, leaf biochemicals, and cellulose and other aspects of plant physiology and chemistry can be absorbed in shortwave infrared. In this example, you see there is large reflectance of near-infrared part of the electromagnetic spectrum in healthy leaves compared to the red part of the electromagnetic spectrum. And these differences in the reflectance of different wavelengths from leaves can be used to calculate different kind of vegetation indices. For example, one can calculate indices to identify healthy leaves, stressed leaves, and dead leaves. And once we have these indices, we can link them with the plant diseases and plant health. There are different types of sensors available, and depending on the need and the objective, one can choose different sensors. For example, for high resolution regular images, there are several cameras available to put on your UAVs. There are also thermal cameras available to look at temperature changes in the canopy and vegetation. There are also sensors available to capture different vegetation indices, and also there are more sophisticated cameras which can capture multispectral data. For example, this camera here shown has 5 sensors to capture data in blue, green, red edge, near infrared, and red spectrum. I divide the data capturing part into 3 additional steps: getting the pilot license to fly the drone, and then a lot of practice of flying the drone, and finally planning the flight. Flight plan has several important considerations that will really impact the quality of data and to get reliable data to reach to your objective. For example, the height of the flight, also speed of the flight, and frequency of capturing the data. How much is the overlap between one image and the next image, and that really will impact your quality of data and then analysis of the data and interpretation of the data. Once you have captured high quality data, now it's time to do processing and interpretations to make decisions for your orchard management. There are commercial service providers which can process and do interpretations of your data, and there are also some open source free tools available which can be used to do processing and interpretations. Some people use hybrid approaches where they take some features of commercial service providers and also some open source tools. Here is an example of an image taken by a drone for this 5-acre orchard. In this one image, you can see the whole orchard. You can easily see if there are any problematic areas in this orchard. For example, here you can see that this area has less vegetation, and to find out what could be the cause, one can go there in this spot and try to figure it out. Is it due to soil? Is it due to disease? Is it due to water standing or some other reason? Now let's calculate the vegetation index for this same orchard. So here in this example, I have estimated red edge index for the orchard. Red in this example means less vegetation. You can easily see the red areas spread across this orchard, but there are some focused areas. For example, here in the same spot, you can easily see less vegetation highlighted by red color. And also, if you look at the lower right side of the orchard, you will see a lot of red spots in that area. So there might be some issue going on in this area because of soil or water standing, and the grower must go there and look and find out what is the cause. In summary, UAVs and sensors can be used for early and proactive pest control, early and proactive disease management. UAVs and sensors can also be used for orchard elevation and drainage management, soil nutrient management, optimization of plant growth, replanting, pruning, and training, and estimation of damage for crop insurance. Now I will speak to you about our work on use of computer vision for automated disease detection in apple orchards. This work is financially supported by Cornell Initiative for Digital Ag. Precise and early identification of diseases and pests is critical for timely management, and we have spoken about it previously. Computer vision can be used for automated, timely, and cost effective disease scouting. However, there are several challenges to implement computer vision for disease prediction and scouting. For example, there are similar looking symptoms of the same or multiple diseases. There could be a wide variation in symptoms of the same disease, and there could be multiple diseases per leaf. A general flow of developing machine learning method for disease detection is, for example, for apple foliar disease classification, first we have to have disease symptoms, then we take images of those diseases, then we need experts to annotate those disease symptoms and those images and develop a data set. Then from this data set, we make a training data set and a test data set. Training data set is the data set where we tell computer that this is how apple scab looks like. And test data set is apple scab, but we don't tell computer that this is apple scab. And using this training set, we train the model for prediction, and then we test this model on test data set. And then we have performance metrics to test the, the prediction accuracy of the newly developed. We have taken a large number of images of apple foliar disease symptoms. This included apple scab, frog eye leaf spot, cedar apple rust, and healthy leaves. We have also done manual annotation of single and multiple disease symptoms per leaf. Then we created a pilot image dataset with all these disease symptoms and then randomly splitted this data into training and test datasets. We have set up and launched Plant Pathology Challenge global competition at Kaggle to develop machine learning models in 2020. Kaggle is machine learning competition platform from Google. It was really exciting to see a large number of teams interested in this competition. We had 1,317 teams submitting more than 22,000 machine learning models. And at the end of the competition, we identified three winners with top models that had prediction accuracy more than 98%. In the growing season 2020, we have expanded this database to more than 25,000 images, and that included more symptoms per disease as well as we increased the number of diseases. Currently, we are doing annotation of these images into distinct disease categories, multiple disease symptoms per leaf, and disease severity percent infection per leaf. And then we are planning to set up Plant Pathology Challenge 2021, a global competition on Kaggle once again. Finally, I would like to present database of apple diseases website. It's an excellent resource and will contribute towards orchard disease management. It has a large number of high-resolution pictures of disease symptoms. It also includes susceptibility ranking for four major diseases of 329 apples. We have also uploaded digital disease fact sheets for 10 diseases, and it has animation videos for fire blight and apple scab. We also have provided a link to YouTube channel from our lab that has videos, animations, and presentations on apple diseases. It also includes brochures, videos, and interactive guides for fire blight detection. Here are screenshots from database of apple diseases website. On left side you see brochures which explain in detail how to use these fire blight detection kits, and on right side you see interactive presentation and a video guide which show you different steps involved in fire blight detection and use of these kits and walk you through every step in detail. This is another example from database of apple diseases website. We have a large number of high resolution images of many diseases of apples. Many of you are familiar with apple diseases from several years of experience, but these high resolution images are very useful for the people who are just starting new apple orchards and also crop consultants and extension educators who want to learn more about apple diseases. With that, I would like to finish my presentation by acknowledging the great support from my team at Cornell Agritech and my collaborators and funding. The funding was from New York Farm Viability Institute for fire blood detection kits and lamp assay, and machine learning research was supported by Cornell Initiative for Digital Agriculture. Thank you very much. And if you have any question, please don't hesitate to write me email. My email address is down there. Thank you very much.